Hey everybody, it's Eugene Lee Show here, and welcome to Forensics Talks. Let me make sure that everything looks good here, because last time I spoke for about 30 seconds and I was on mute. There you go. That's typical of me. Looks like everything's good. Just a couple of things before we get started. First off, I want to welcome everybody. Hoping you're having a good day. This is Remembrance Day in Canada, so I just wanted to bring that up for everybody. Don't forget that we are live streaming to YouTube, Facebook, and LinkedIn. So hopefully you're catching us on one of those particular platforms. Also, everybody should know that if you are watching this on YouTube, there's a comments section. So if you type into the comments, it'll actually pop up here on my screen. So if you have any questions today, anything you want to say or anything you want to comment to, by all means, please put it into the comments section, and then I'll try to get that to our guests here. And I often forget, but I'll make sure that I look over every once in a while. Also, let us know where you're from as well. I always like to know when there are people who are international here. If you're from South America, if you're from Europe, wherever you're watching, it's always nice to see. And I already see Gail saying hello. So hi, Gail. Nice to see you. Okay, just very quickly, there are just a couple of quick announcements here, and the first one has to do with the Forensic Photography Symposium. So this is going to be held on January 17th to 20th, 2022. And if you're interested, this is a symposium that's really focused on forensic photography, okay? Really just looking at people and some of the problems that they face when they're using a digital camera to document evidence. And that could be for crash scenes, it could be for crime scenes, it could be for forensic anthropologists, photographing bones and all kinds of different things. So we're already having really great success with the amount of speakers and presenters from all over the world. So if you are interested in attending, registration just opened yesterday. So you can just head over to ai2-3d.com.fps or /fps, excuse me, and you'll find all the information there, registration and everything else. Also, if you are interested in submitting an abstract, there's still a few slots open, and by all means, there's an online form there. Put in all your information. It'll force you to upload a headshot and stuff like that, and we'll receive that and review as well. Also, I want to say that there is another conference that haven't talked about it just yet, but it is the International Association of Forensic and Security Metrology. It's a mouthful, iafsm.org. They are having a joint conference with the Association of Crime Scene Reconstruction, and you can see here it's February 14th to 18th. If you head over to iafsm.org, it should be a good conference. It's like a 2-for-1 because you're getting a mix of a whole bunch of different people working in crime scene reconstruction using 3D technologies. And the real cool thing is that on the Monday, well, there's a couple things going on, but on the Monday in particular, there is a mass casualty event. So I believe there's an arena, and people are going to be simulating like a shooting, and it's going to be pretty dramatic. So people are going to be there with all their tools and documenting stuff or whatever. So it should be a fun time. There's also a basic laser scanner certification course, and I'm teaching a good part of that on the Sunday beforehand. So if you are using 3D technologies like a laser scanner, we can definitely, you can do the basic course there and get a certification while you're at the conference. Excellent. Okay, so let's not waste any more time, and let's start getting into this here. So this is episode 48, and my guest today is Dr. Amoret Whitaker, and she's the program leader and a senior lecturer at the University of Winchester in the Forensic Studies program. She has a Bachelor of Science in Zoology from the University of Reading, a Master of Science in Taxonomy and Biodiversity from Imperial College London and the Natural History Museum, and a PhD in Forensic Entomology from King's College London and the Natural History Museum. Dr. Whitaker specializes in forensic entomology, and since 2004 has undertaken forensic casework throughout the UK for many police forces and forensic providers, attending crime scenes, post-mortems, and attending court if required. She's done research in areas related to forensic entomology, taphonomy, lots of blowflies we're going to be talking about today, and maybe we'll slip in some stuff about fleas as well. Her teaching responsibilities are mainly in forensic biology, trace evidence, forensic medicine, forensic imaging. I know she does some photography because when I spoke to her last time, she was actually teaching photography to one of her classes, research methods and reporting forensic evidence. So let me get her in here. Good evening, Dr. Whitaker. How
Hello.
are you?
Hi, good, thank you. Yeah. Thank you for inviting me.
Well, I realize this is your evening, so I appreciate you taking some time out of your day to spend time with us. So, well, the first thing that I always ask people, because I'm always interested in, is how, before you were Dr. Whitaker, before you got into this whole thing, were you the kid that was digging in the dirt and looking at stuff, or were you doing something totally different?
Um, I wasn't really digging in the dirt, I have to say. I'm not one of those kids who kind of grew up collecting insects and things. No, I mean, I grew up loving animals, and actually it was doing a zoology course at the University of Reading, as you said. And I kind of did that because I thought that I would come out and save furry animals. I'd work with gorillas or polar bears or chimpanzees or, something cute. And actually, the first time I looked down a microscope at insects, I was just absolutely hooked. And I just didn't know the diversity that we had, and everything after that was just all about insects.
I
So
heard,
all my essays, dissertation, projects,
Mhm.
whatever. Yeah.
I heard you say that before about, you were looking down the microscope. So I was curious about what did you see that made you go, oh, it's, and a light went off. What happened?
I think just the detail, really. I mean, obviously, insects come in all shapes and sizes. Most of us don't really pay a huge amount of attention to them unless they're kind of like the big showy ones, the dragonflies, the butterflies, which are lovely. But actually, when you look at something like the humble housefly, it's just fascinating. And what I love about teaching, and teaching entomology especially, is that I see that same reaction from my students. So they start off kind of going, oh, I hate insects, I hate maggots, blah blah blah. And then I'm like, okay, look at it down the microscope, and by the end of a 2-hour practical, they're just completely hooked. So
Yeah,
yeah.
I think they're quite. I think they are. I can see the same thing. There's somebody online, or somebody who I know, who posts micro CT scans of insects, and they're absolutely fascinating to see when you're down at that very small level. Sort of just the bodies, the structures, the arrangements that they have. So yeah, I totally get that. I'm kind of fascinated by that as well.
Right.
I want to ask you about. Well, let's get right into it. Like, in terms of forensic entomology, what would you say is the main role or the main goal of forensic entomology in crime scene cases, in different types of cases that you've worked on?
Yeah. I guess the kind of obvious one is assisting with establishing time since death. That's the one that we tend to hear about. But actually, it's really there's so many cases where insects are involved in some way or another, and actually all it needs is an expert in those particular types of insects to actually be able to shed some light on the investigation. And it can be something really, really simple. Yeah, I mean, I tend to sort of think that most of the cases I get involved with are estimating minimum time since death. But actually, a few years ago, I kind of decided to look back at the previous 100 cases we'd done. And actually, I was amazed at how, the huge variety of cases that there were. When I say we, I mean the Natural History Museum. And it wasn't necessarily all about deaths and murder. There was a whole bunch of other things that people want to know.
One thing that I was wondering about before, so you were referring to the post-mortem interval, like time of time
Mm,
since death, but
mm.
are there other things that the type of insects that are on the body? So if there's like more of one type of insect versus the other, I don't know. Are there other small details that you can pick up from just the insects that are there on a decomposing body?
Yeah, I think it's really important to kind of not to look at these things in isolation. To kind of consider the whole scene,
Hmm.
and it's really important to understand the ecology of insects and different species as well, so that you can kind of put together a sort of picture that shows the events that took place. So crime scenes can be quite complicated, and you certainly never ever get the same crime scene twice. They're always completely different. So there's lots of things that you can determine with insects, and a lot of it depends on the type of insects. So obviously, if you're talking about minimum time since death, then the blowflies are going to be the most important insects.
Yeah, I was just going to ask you. I was going to say blowfly is the one that comes up in all the papers, like everybody's researching that. But what can you say about the variety of different insects that could potentially present themselves? Like what are maybe some of the most common and maybe some uncommon ones that you've seen before?
Well, when a person or an animal dies, they obviously start to decompose straight away. And I apologize if anybody's having dinner or lunch, because my talks always seem to be, around meal times. But you get this succession of insects that's associated with the different stages of decomposition. So as the body starts to decompose, it gives off different odors, and of course it changes physically as well. And so you get different insects that are attracted at different stages. The reason that we really focus on the blowflies is because generally they are the first insects to be attracted to the body, and therefore, if you're trying to estimate time since death or minimum time since death, then those are the insects you want to focus on.
Okay.
But there may be situations where a body is in a different situation. I was listening to the discussion you had earlier with Paola Magni, who I know very well, and of course she deals in aquatic crime scenes. And so there's all sorts of information that you can get from insects. You ask what's a kind of insect that you don't often see. You mentioned fleas, which was, is also a kind of specialism of mine. And actually the first case I ever worked on, and the only one I've ever worked on that involved fleas. Really, it literally just took having, knowledge about the ecology of fleas to be able to contribute to the investigation.
Interesting.
And it was I wish all cases were that simple, because it really didn't take any complicated analysis or anything. It was just, this is what fleas do, and this shows that the suspects are not being truthful. So and that was enough.
The blowflies are they fly, so they get around really easy, and that would seem to me one of the simple answers as to why they're there first. But things that I've seen before in some of the research that you've done, like for example the beetles and other types of insects which crawl, is part of the reason that they're waiting to get there. And the question I had actually beforehand, before I forget, had to do with when you talked about decomposing bodies. What is it about the decomposition? Is it something to do with, is it a chemical that's released? Is it an odor? Is it both? What is it?
It's both, really, and obviously it's the breakdown of tissues as well. I'm not a chemist, so I leave that part of it to other people. But you definitely see very clear physical changes as the body decomposes. And of course, the difficult thing is some of that is due in part just to the body kind of breaking down, but a lot of it is actually due to the insect activity as well. So, and obviously when you get the two together, you can get very, very fast decomposition. But you mentioned beetles. Obviously beetles can fly. They do have wings. The thing about things like beetles is they tend to be dependent upon the blowfly activity, and more specifically the larvae that are already on the body. So that's why you can't really use beetles as a kind of good estimate of time since death, because actually they're dependent largely on the first colonizers, which is generally the blowflies.
When you say they're dependent, do you mean that they use them as a food source, or because there's just something else going on?
No, they use them as a food source. You actually get lots of insects that, there's sort of different types of insects that have kind of inhabit different ecological niches. So you've got the things like blowflies, which are necrophagous, so they are feeding purely on decomposing animal or human material. And then you've got things like predators and parasites, which will actually be attracted to the body mainly because of the insects that are already feeding on it. And then you'll get adventive species, so things like woodlice and spiders, which actually are just making use of the kind of physical structure of the body, and they have no forensic value at all.
Mm-hmm.
And you also get omnivorous species, so things like wasps and ants, which will feed both on the body itself, but also they will feed on blowfly eggs and larvae as well.
Okay.
So it can be sort of quite complicated. You can't really just look at one thing in isolation. You have to kind of look at the big picture.
Yeah. So temperature is actually a big factor here, right? So in for example, in the middle of summer, and you have a body that's whatever, somebody's been murdered in the middle of a field or in the middle of a forest or something like that, versus cold winter and they're someplace else, that's going to have a huge factor on the insect activity.
Absolutely. Yeah.
What other kinds of things can have, like, so I'm imagining there's all things popping up in my head. So for example, what if somebody tries to cover a body or wrap a body in a bag or like a blanket or something like that, where it's not so easy to enter or deposit the eggs? And I'm sure those things have a way of finding their way into everything, because when I eat my you have a banana on the desk for like 5 minutes, there's a fruit fly somewhere, that pops up. But they tend to be very prolific. They tend just to show up sometimes. So I'm amazed sometimes at how quickly they get there.
Yeah, I mean, things like flies are ubiquitous. We don't see as many of them in the wintertime, but for sure, they are around. There may be fewer species, but yeah, certainly if somebody, drops dead, or an animal or whatever, then you are going to get normally blowflies feeding on the body and laying their eggs on it. I mean, you mentioned things like sort of wrappings and stuff, and accessibility to the body is a big issue. That's something that we really have to take into consideration, because there is a massive difference between a body which is, in the height of summer in the daytime, which is, somebody drops dead in the middle of a field, for instance, or a wood, and their body is going to be colonized pretty quickly, literally within minutes or so. Where you're looking at bodies that are inaccessible in some way, so maybe they've been wrapped or they've been buried or they're indoors or, something like that. Obviously, it's going to take a longer period of time for the blowflies to find the body, and therefore for the females to lay her eggs on it. So you really have to take those things into consideration. And that's one reason why it's actually really important, I think, for an entomologist to go to the scene. Because then they can really, assess the whole scene. It can be a lot harder once the body's been recovered, and if someone else is doing the collecting, then you can never go back to the original state, and you have to sort of rely on photographs and things. I mean, blowflies are incredible how they can get access to bodies. Certainly things like bodies indoors doesn't stop them. So say if I go to an indoor scene, one of the very first things I'm thinking about is doors and windows, and were they open? Could the insects get easy access, or is there lots of, detritus and rotting food lying around, in which case there's probably blowflies in there already. So they are incredible insects.
You had mentioned once, I heard you mention once, that the more decomposed, like you mentioned, going to crime scenes, so the more decomposed the body, the easier it was for you to deal with. Did you mean that in the sense of like dealing with the fact that there's a dead person there, or did you mean it also in terms of the work? Is there something easier in terms of dealing with the insects, or something in terms of it makes it easier for you in terms of the work that you're doing?
I think in the sort of context of when I've said that, I've meant it in terms of the you know, the kind of emotional, like how I handle going to scenes, because you know, I'm actually quite a kind of squeamish person, really. So what I tend to find is if you, you know, if you're dealing with a body which is, you know, hasn't been deceased for a very long period of time, it's relatively fresh. To me, you know, it really looks like a person. It looks, you know, they could be asleep almost. Whereas the more decomposed a body is, the less like a kind of human being they look like, and therefore the more kind of detached I find I can be. And obviously, you know, I kind of do get very excited by large maggot masses, which we, you know, in this country, we don't get them very often. We only get them kind of in the height of summer, really. So if I go to a scene and there's a very decomposed body and there's, you know, big larval mass on it, then yeah, I get very excited about that.
Okay, that's good. That's funny because so like, you know, we do things like with blood stain pattern and people are like, you know, you're getting excited about blood, like, you know, it's not something typical.
I know.
There's something wrong with us. Yeah, I know. I had a question. Could you explain the basic process, sort of like the life cycle of what's happening when, you know, Blowfly first encounters, you know, a dead body, and then how you kind of go through like to the larvae stage and stuff like that?
Yeah. So as I said, when somebody dies, they start to give off odors, and you will get blowflies attracted to the body and feeding on the body. And the female blowfly needs a protein meal in order to produce eggs. So quite often they're flying around with eggs already inside them, ready, developed already, ready to lay on the next body that becomes available. You will also get males kind of hanging around, but they tend to be sort of on the outskirts, you know, ready to mate with any females that turn up. So the females will feed on the body, and then she will start to lay her eggs on the body. And generally, she will lay her eggs in areas which are kind of moist and dark and hidden away. She doesn't want her eggs to dry out or to be predated on. So more often than not, with humans for sure, and especially if they're clothed, then obviously the facial orifices are going to be available. So that's one of the first places that you will find the eggs. But also places like the hairline, around the back of the neck, sort of on the, you know, between the body and the substrate that it's lying on. These eggs will then hatch out into very small larvae, so what we call first instar larvae, and they will start to feed on the body straight away. So this is also why they need to be laid in an area which is nice and wet and not dry, because they won't be able to feed. The larvae will feed and they'll grow, and they go through two molts as well. So they go through first, second, and third instar larvae. And then, so this is the kind of main feeding stage. Once they've finished feeding on the body, the third instar larvae will then move off the body to pupate. Now I say that, but there's one particular species that we get in this country, in the UK, which actually pupates very close to the feeding source. So that makes life really nice and easy when I go to a scene and I find a kind of nice clump of puparia just sort of, you know, just below the head region. But otherwise, they tend to move off the body, and they can wander for quite a long distance. It depends very much on the substrate. So if it's outdoors, if there's a lot of earth around, they might burrow down into the soil or under leaf litter, etc. If it's indoors, they'll go under furniture or under carpets or under skirting boards. And so sometimes they can actually be quite difficult to find. But then once they've found a suitable place to pupate, the larva will constrict and basically start to dry out, and the outer cuticle would start to harden, and that becomes the pupal case. And then within that, the rest of the larva will basically break down and over time it will become an adult fly. So it's an incredible process that, to imagine that, to think that a maggot can become an adult fly. It's amazing, really. And then eventually the adult fly will emerge from the pupal case, and the life cycle will start all over again.
Oh, so I want to start right there. So it starts again. So does that mean you can have many generations of blowflies?
Generally not on the same body. And the reason is because the blowflies are attracted to the body at an early stage of decomposition. By the time you've, depending on the environment and the temperature and everything else, by the time those eggs have developed as larvae and gone through the whole pupal stage and emerged as adults, the body is probably by that point not suitable for blowflies to lay their eggs on. So either there's not going to be a huge amount of kind of wet material, wet tissue remaining. It may have mummified, for instance. It may just be skin and bones if it's a small carcass. So it's a nice idea that you'll get like lots of generations all, developing on the same body, but in reality that doesn't tend to happen with blowflies.
I see.
It can happen with some flies. So there's a type of fly called the coffin fly, and they have been known to go through a number of generations, literally underground in a coffin, because that sort of environment suits them.
Oh, interesting. So when the female lays eggs, about how many eggs is she laying?
Do you know what? It's one of those things that we actually know very little about. You would think with our most common species of flies that we would have all this information, and it's something I actually got one of my students to research last year. Unfortunately, because of the pandemic, she wasn't able to do. I wanted her to do field work and lab work, and that didn't work out. But we read, there's so many publications out there, and they all give these quotes. They all say, this particular species lays 80 to 100 eggs, or 60 to 70 eggs, or 100 to 120 eggs in a batch, or whatever. But actually, as far as I can see, nobody's really tested it. Nobody's researched it. So we're all kind of quoting the same things, and we really don't know. Just these really basic facts we just don't really know the answers to.
Yeah, well, the reason I asked was because in some of for example, in some of the research that you've done, and I've gone through some of the papers, and I'm looking at the images, there are clusters that look like there are, I don't know, thousands of these things. So I don't know how many female flies actually made it there, but I mean, have you seen, well, let me ask you, like, how many estimate of how many, larvae you can have, or how many eggs can be laid? I mean, we're talking.
It is thousands. I mean, I've certainly seen what you quite often get is these kind of aggregations of blowflies. So where one female blowfly started laying her eggs, then others will come and they'll all start kind of laying on top. We're not 100% sure why this happens. There might be a number of reasons. So it could be to sort of reduce the chance of predation. So there's kind of safety in numbers. Also, because it's a good site to lay. Perhaps because there's less chance of them drying out or desiccating if there's lots of them. So, I mean, the biggest egg mass I've ever seen was actually when I was doing my PhD work out in Tennessee and using human donors. And I put the body out kind of in the morning, went away, for a few hours, came back, and literally there was this egg mass that was the size of a tennis ball on the head. It kind of started in the nasal area and was massive. And unfortunately, there was a massive rainstorm that night. And so when I came back the next day, a lot of it had just washed off. And a lot, probably drowned. So, I mean, this is the thing, that there's so much we still don't know about the humble blowfly.
There's something else that I had read I thought was quite interesting, and that was, and you wouldn't think of it because of the mass of a a pupa or fly or whatever, or the larvae. But so a body that's decomposing and that has, let's say, a mass of these eggs or whatever, or larvae, the larvae affect temperature as well. The clusters have
Yeah.
sort of generate their own heat. Is that true?
Yeah, that's right. When you get these big larval masses all feeding together, the kind of friction of them basically increases the local heat, and it could be by a huge amount. So I've done quite a lot of work both on stillborn piglets in the UK, but also on human cadavers in Tennessee, and it tends to happen towards the end of the second instar stage and throughout the third instar stage. So it doesn't happen with eggs or with the very small larvae when they've just hatched out. But in the UK, I mean, even just a very small larval mass increased the temperature sort of from ambient to larval mass temperature by about 4 degrees. In Tennessee, I was seeing like a 20-degree, so we're talking centigrade here, 20-degree increase, which is massive.
That's huge.
So, I mean, it's really important to sort of recognize this because, as you said earlier, temperature is all important. So, when I work out how old the insects are, I have to try and estimate what temperature they were developing at. And so you have to take this larval mass increased temperature into account.
There was, I believe there was sort of like an upper limit, like I don't remember if it was 47 degrees or something around there.
It's around 42 degrees, I would say.
Okay.
Yeah.
Okay.
Yeah.
So if you're in an area like, for example, southern, like you're in Arizona near the border or near the equator or something like this, where temperatures can exceed that, do you find that it actually decreases? Like you get less? I was just wondering if you get in very, very hot environments, it becomes, I don't know, less of more of a problem.
Yeah, I mean, I think for a start, the populations are probably better adapted to higher temperatures. So you do get differences between different populations in different countries. But something that I also found is that, if it's very hot, the larvae they'll tend to kind of hide away. I mean, they won't be kind of in full sun. So, for instance, in Tennessee, I did quite a lot of thermal imaging. And what I found was that where you had cadavers that had quite a high fat content, and if they were in full sun in the middle of the day, the surface of the body was actually very, very hot, and the larval masses were actually cooler than the surface of the body itself. So, this is why it's so important to take temperatures at the scene. So to take temperatures of the skin and to take temperatures of the different larval masses around the body so that you can try and determine, the temperatures that the larvae are actually developing at. And what they also tend to do is they tend to, you'll sometimes get these kind of writhing masses where the larvae will move in and out of the larval mass. So the center of the larval mass is very hot, and you get these kind of, the outer margins get cooler and cooler. And if the larvae stay in the center of the mass for too long, they will overheat and they'll die. And so you can see with a thermal imaging camera that the larvae are literally kind of going around in circles, and they're kind of moving out of the larval mass, cooling down a bit, and then coming back into it. So, it's fascinating to research.
Yeah, interesting. I mean, you've been doing this for some time now, so I wanted to ask you about, for example, what have you noticed, or what have been some of the major changes in forensic entomology from when you first began versus today? Like, what's sort of been some of the more notable things that have improved the accuracy or the study? What kinds of things were sort of big improvements over the years, or discoveries?
I think there's more people doing research, which is really important because, as I've said, there's so many basic things that we just don't have the answers to, and so it's really important for people just to do kind of basic developmental research on different species. So there's a lot more data out there now. I think there's a much better understanding of the subject by police forces and forensic students who were obviously going to go out and they're going to be working in police forces.
Mm-hmm.
So it's, I think back when I first started, if somebody had asked me what I did, they wouldn't have, even just the person on the street wouldn't have a clue what it was, what it meant.
Mm-hmm.
Whereas now most people have heard of it
Right.
from somewhere. So I think, you kind of have to explain yourself less, really. People kind of understand the benefits of it. And I think standards have improved as well, which is really important. And that's the same for across the board with all forensics. It's really important to have kind of standard protocols that everybody sticks to, so that, we're all kind of, keeping up with the latest research.
Mm-hmm.
And so I think definitely standards have improved, and just general knowledge.
You have to go to, I mean, you testify once in a while. You have to go to trial and talk about, what you found or whatever. Do you find that more times, people are rather accepting of forensic entomology, or do you still find you're on the defensive at times, or what are your experiences at trial?
You can be on the defensive, because obviously if I'm appearing for the prosecution, then if the defense have got somebody in to argue their case, and if they don't agree with my report, then yes, of course you have to sort of defend your evidence. I would say obviously with some some areas of forensics, such as pathologists, for instance, they probably go to court for pretty much every case they work on, whereas I find I probably get called to court about 1 in every 10 cases I do. So it's not a sort of particularly regular thing, and I think the reason for that is either because the courts accept my report and they don't require me to go to court and explain it.
Mm-hmm.
More often, though, I think it's because entomology can be really helpful in the early stages of an investigation. So, especially if somebody's, say, been missing for a long time, apart from the obvious question, like who is the person, one of the questions the police want to know is, when did this person actually die? Like, do we need to be investigating, 6 months ago when they first disappeared, or is this something recent? Have they died very recently? So
Right.
Quite often what entomology can do is that we can give the police a good time frame to work within, to investigate. So if I say to the police, this person's definitely been deceased for 5.5 months, say, then they can focus their investigation on that period of time. They don't have to look over, the last 6 months of CCTV footage or digital evidence or whatever it is.
Yeah.
And that means that quite often then they will then get other evidence which, will support my results, which is that, the person died 5.5 months ago. And so then maybe they get other evidence which is actually more compelling than mine. And so they actually don't need me, they can just present my report at court, and they can say, based on the entomologist's report and interpretation, then, this is the time period that we investigated, and this is all the other evidence, supporting evidence that we found. So thankfully I don't have to go to court very often, because it can be, it's
Stressful.
Yeah, it is
Yeah.
stressful.
Yeah, I totally get that. Trust me. I know all about that. I've got one coming up next week, but oh well.
Right.
So here's the question. Phone rings. It's, hey, we need you to come to a scene. What tools are you bringing with you? What's in your toolbox? What's in your tool bag at the scene? And what kinds of tools or hardware or whatever are you using back at a lab?
It's actually fairly basic, really. It's forceps. It's so all sorts of different types of forceps. I have some fabulous forceps that I bought in America that are about that long, which are great for going down the backs of sofas or into bins or whatever, and I don't have to get too close. Lots of vials, tubes to put the insects in. Hot water and ethanol for preserving them. Data logger is really important. So we've already mentioned temperatures, and so it's very important to put a data logger at the scene to record the temperatures at the scene. Yeah, that's about it really. And maybe some cat food. So I raid my cat's larder, because if you're collecting live insects, you want to preserve some if they're still feeding on the body, and you want to be able to rear some of them through to adults as well. So they need something to feed on. And you obviously don't want to be chopping bits off the body. So I tend to take along a pouch of cat food.
So, and when you're collecting, are you collecting just the insects themselves? Are you collecting other things as well? Like, what kinds of things are you bringing back with you?
Really the insects, so adult insects, if there are any. So, say if there are insects still interested in the body. If you're indoors, for instance, you may have had the adult insects emerging from the pupal cases. So you may have, quite often bodies are found indoors because a neighbor reports lots and lots of flies. And that's quite often because they've, gone through a full life cycle, and then obviously hundreds or thousands of flies have emerged, and then they're all at the window, or they're going out into the corridor or something. So I collect adult flies as well. It's really important. One tends to focus on the body, but the oldest insects may not still be on the body. So those are the post-feeding larvae, so the ones that have finished feeding and they've moved off the body, or the pupae, or indeed the empty pupal cases as well, because with very common species of blowflies, you can actually you can identify the empty pupal cases to species as well. It is mainly the focusing on the insect activity. Photographs, obviously, I take. So sometimes there's usually somebody who's been assigned to do the photography, but I quite often take my own pictures as well, because I know specifically what I need to take pictures of, and it may be really things that other people wouldn't think about. Like I might take a picture of the windows or the skirting boards or the doors to check for gaps. Yeah.
There's actually, I just want to show people here if I can, but this is a good little article. I thought I was going through this. There's some really good information here, and I'm just going to paste this. This is available online. So for those of you that are watching, I've just pasted the link there, and I thought it was a nice overview of some of the considerations in forensic entomology.
If you go back up to the picture at the top, that very unglamorous person on their knees going through piles of dirt is me.
Yeah, okay.
So this is what I show to my students at the start of the program, and I say, if you thought this was glamorous, think twice, because it's not.
Yeah, it's a good point. Yeah, I often try to. Even yesterday, I had a guest lecturer in one of my classes, and it was great because he was trying to convey, look, he goes, we're dealing with pretty messy and nasty situations, so you really need to have the emotional well-being and the mental fortitude to handle these kinds of situations at times. So it's a very good point.
That's that's a big thing at the moment, because actually there's a lot of counseling available for paramedics, people that work with the firemen, police. So if they have to attend a train crash or some sort of mass disaster or something, but actually forensic practitioners are dealing with cases every single day, and there's actually very little in the way of sort of support for the things that we have to deal with. So that's much more sort of considered now, which when I first started it wasn't even a thing really.
Yeah, definitely. I've been seeing that come up as a presentation topic at conferences more often now than before. Yeah, that's true. Yeah. I want to ask you about, I thought this was interesting. You did a paper on the effect of burning on
Mm.
the minimum post-mortem interval. And I thought that was really
Mhm.
interesting because there was a couple of things in here that were little surprises for me. But do you mind just maybe just going over, just giving a quick overview of what you did here?
Yeah, so this was a review article really, based on a few cases that I've attended, or that my colleague at the museum has attended, and also looking at research that people have done. And actually, very little research has been done on the effect of burning on insects and our ability to estimate minimum time since death. And I have a PhD student at the moment who's looking at this very thing. But what I've found, so I've attended one particular scene where somebody had died, and I think it was determined that he had sort of died a natural death. But sometime after that, the sort of, he was living rough, and so the shed where he was kind of living was set on fire by some kids. And some poor fireman, I think it was only his second day on the job, and he was given the job he was given a very long rake to basically rake over the ashes. And he managed to flip over this body and actually separate the torso from the legs.
Mm.
And we could kind of see very clearly from the burns on the body that the body hadn't been in two pieces, because half the body was actually, the part of the body that had been lying flat on the ground was actually protected from the flames. And despite the fact that this whole shed had gone up in flames and it just looked like a complete wreck, there were still live larvae feeding on the body. So I was able to give a very accurate time since death, despite the fact that it was a fire scene. The other thing that you find is that you may have bodies. So quite often people will try and conceal their evidence. So they'll kill somebody and they will set fire to the body, quite often using accelerants. And so the question we were trying to answer is whether insects, specifically blowflies, will still be attracted to the body. And what we found is that if they tend not to be attracted to the body if it's just kind of charred, so sort of smoke damaged. But if the body burns a little bit longer, it tends to split open. And what this does, of course, is to reveal, fresh, unburnt flesh. And so you'll get insects or blowflies laying their eggs, within those cracks in the body. And so it doesn't prevent insects necessarily from colonizing the body. So I think, this is the kind of information that it's important to get out to the police, which is just because you've got a fire scene doesn't mean to say that all the evidence has been destroyed, specifically the insect evidence.
Yeah, that's really interesting, and that's like the key there. You also talked about there was like different classifications in terms of the burning, like what right
Yeah.
from sort of death from smoke without heavy burns, but right up to like cremation, like where everything's just sort
Right.
of So
Right.
I don't remember where that was on the scale, where you sort of have the body sort of breaking.
It's called the Crow-Glassman scale, and it's a paper by Glassman and Crow, and they actually kind of devised this sort of range of burns specifically for deceased remains, because we already have it for living people. We have sort of first, second, and third degree burns, but the same thing didn't exist for deceased burn victims. And I think they devised it after the Waco disaster I think it was in Texas, many years ago. And it's basically a way of being able to describe the level of burning so that you can communicate better between, paramedics, the fire department, the pathologists, the scientists, etc. So CGS1 is kind of smoke damage. CGS2 is where the skin starts splitting. CGS5 is basically cremated. Yeah.
Interesting. So I want to ask you about sort of the where forensic entomology is in terms of progress. So, for example, I mean, you've been doing this for a while. So, are you starting to see things? I don't know how to describe this, but maybe converging or funneling in on a particular position, or do you find that the further you go, you're actually there's more opportunity to do a whole bunch of other things that have to be investigated and more has to be done. Which way do you see it going for you?
I think without a doubt, we need more people doing it. I mean, in the States, there are more people, but then obviously it's a much bigger country. So probably by land mass, we have the same number of people per square mile or whatever doing forensic casework. It's not really a full-time job. So, I do get students saying, I want to be a forensic entomologist, but, unfortunately, no police force or forensic provider is going to employ a full-time forensic entomologist because there isn't enough work out there. Because although we hear about a lot of murders and stuff like that, I mean, thankfully they're relatively rare. And entomologists don't tend to be required with, the kind of gang stabbings in the street, where clearly somebody's died just a few hours before they were found. So, we really need more people like me, really, more, university academics and researchers who are doing this, as I was going to say extracurricular activity. It's not really like a hobby, but as part of their job. So that we can kind of cover the whole country. So that, for instance, if I'm called out, quite often I might be called out to Scotland or to, north of England or down to Devon or whatever, and it might be 5-6 hours journey to get there. And in an ideal situation, we would have people all over the UK who are focused on their particular area, and they're used by their local police force. That's where I'd like to see it going, and more research, much more research being done. But it's a difficult topic, really, because it kind of falls between so many gaps. It's not really medical. It's not really ecology in what we sort of think of as being ecology. When we think of things like biology, we tend to think of, either big animals or kind of blood and genetics and things. So forensic entomology tends to kind of fall in the gaps when it comes to funding. So it's actually quite difficult to get grants and to get studentships, for PhD students.
Right. Question. Just a couple more questions for you, but one is, what kind of topics or what kinds of things are you focusing on right now or in the future? What are sort of the areas you're really honing in on?
I think, one is the burning, which my current PhD student is doing. Another is, as I say, simple things like how many eggs does a blowfly lay? How does that differ between species? There are quite a few species that do crop up more and more in casework, and we don't have any developmental data for those at all. So it'd be really good to be able to spend the time just doing basic developmental studies of, some of our most common species.
Yeah. I'm not sure.
Yeah.
I'm just going to show you here. I just noticed that Helen here is saying
hello. Helen.
And she's just saying that she's trying to convince the Ontario the Forensic Pathology Service to look at a full-time position since they now have a forensic anthropologist. So I was actually thinking when you were talking about that forensic anthropologists are somewhat in the same boat, that it's difficult to find a lot of full-time anthropologists.
I think anthropology is probably kind of more used because whenever remains are found, you actually need somebody to say yes, it's a human and it's not a dog or a chicken or whatever. So perhaps there's a little bit more work available for them.
Yeah, absolutely.
Yeah.
Well, I got one last question for you. And a long time ago, I used to have a Yamaha motorcycle, and apparently you have one that has a special name. Why don't you tell us about that?
I haven't been on a motorbike for a long time. So yeah, my first motorbike, which was a little 125, was called Hister, after Hysteridae beetles, because she had a beautiful black shiny tank like those beetles. I have another motorbike at the moment, which is a ER 500, which is it's got a lovely green tank, green color, so that's called Lucilia. Yeah. So yeah, I tend to name motorbikes, chickens, all sorts of things after insects. Yeah.
Why not? Excellent. Well, I'll tell you what, I think we're going to leave it off there. There's some great information you have. I did want to say also for those that, let me just see here. Let me sure if I can share my screen again, because if anyone was interested and you want to get hold of Dr. Whitaker, she is, if I can find it, there we go, on both LinkedIn, and let me just bring that up here. So she is on LinkedIn, and she is also on here. I'll take this address and I'll just put it into the chat window, because you can also find her at the University of Winchester.
Just Google me and it'll come up. Yep.
She's got her own Wikipedia. She's got stuff.
I don't know where that came from. I mean, you're not allowed to set up your own Wikipedia pages, are you? And somebody did that.
Well, somebody's taking care of you. That's for sure.
I don't know who.
That's good. Well, listen, thank you very, very much. I really appreciate it. Hang back for a second, and then I'll come back and chat with you in just a bit. All right, great.
Thank you.
All right, folks. Well, that does it for this particular one. And so I want to say thank you to everyone here. Don't forget, we do have a couple of things. One was the Forensic Photography Symposium, and I talked about it at the beginning. I'll put in the link in the comments section once we're done. And also don't forget about the IAFSM conference as well, if you're interested. Thanks very much. I wish you all a great November 11th and hope to see you again next week. Take care. Bye-bye, everybody.